I'm Vladimir, the Russian biohacker, living and working in Belgium, just like Vladimir used to do it a long time ago. And I would like to present you my long time ago invention. It's called Frankenstein kit.
Frankenstein kit?
I'm working in biohacking and DIY bio, do-it-yourself biology. So what is about? The major thing, it's all about the interspecies genetical communication. We all have genes, and in fact the human genes are not that different from plant genes or insect genes or and other animal genes. So it's possible to transfer genes between species using the modern scientific methods. And the Frankenstein kit is all about this. Like, experiment with DNA. Try to get how to get another species DNA into your body. So the kit consists of basically two parts. One part is how do they do it. So you can make your own DNAs from any samples, biological samples you find interesting. And there is a basic part from 9 samples here that contains a already prepared DNA by me from different species. The number 1 is the Sinmaharius from bone remains. It's from Catholic Church, from Reinau DeVos, my friend. It has a provenance, it's proven that it's really old saint DNA. And then I have a ship, the Dolly. Not many people know that from the Dolly a piece was taken when she died and divided into a few private collections. And one of those private collectioners provided me a small sample from his piece of Dolly that I used to extract DNA. And then we have a horse from Russia, which is a cryptic one because I cannot tell which one horse is this. It's very famous horse, but it doesn't matter. And then there is a rat, Amadeus the First. It's my pet rat, Norwegian black rat. And yeah, it's a piece of tail of it. Died 3 years ago. And then we have a potato, DNA from the experimental fields, also private collection, because the people were really jailed almost for destroying that field. And then there is oak. It's an 800 years old tree from Romania, from the Sighișoara forest, almost next to the birthplace of Vlad Dracula, where I stayed for some time to pay tribute to my relative, and to also I visited that ancient forest and I talked with that oak that so still alive, 800 years old trees, yeah, still alive and okay to live no longer. And then number 7, I have my own DNA in the kit. It's Vladimir DNA. And then number 8, it's honeybee, which I found on the counter in Ghent. That one, yeah. And the number 9 is a spider from Japan, Tsukuba City, where I used to work for 4 years, long time ago. That spider is very special story, and it's going to play later. I walking every day to my work and back to the home. The name of the house was Mayfair, yeah, and I walked through the Doha Park, yeah, to my frontier research lab. And once I saw the spider making his net, Japanese spider, really big one, and very strong net. And every day I was walking next to his net and I saying hello to the spider maybe saying hello to me, and we became friends. For 3 months I was watching the spiders, and typhoon season came, and after every typhoon, net is gone and spider working, remaking those net. And I was like, yes, it's a quality, very high quality Japanese spider net. Yeah. I was so much surprised and so much appreciated. At some moment I got a zen there. The zen came on me, ascended on me. And then I just took the spider and put it into the ethanol, 100% ethanol, and preserved it for until last moment. It was always with me, my best friend. I was always talking with the spider. And then when I invented that kit 2 years ago, under influence of Liz Parrish from BioViva, that spider DNA, I extracted from that spider that was my friend for many years, that spider. I extracted DNA, and then it's number 9 here. You can see it. This is the DNA of my spider. Yeah, that's a piece of it. I have much. I have bigger volume. Yeah, that spider DNA, I'm going now to inject into my body. So what is going to happen? Well, I don't know. I just want to revive my old friend, my spider, from Japan, because that DNA is going to come into my body, and then it will meet my cells, human cells. I can afford to call myself human because I live on the earth for a very long time with humans, and I would like to see what will happen. Like that DNA will go into the cells and make an interaction, communication with my human DNA. So how actually it happens? That kit also provides opportunity for the people to make their own DNA of choice. So what we have here, it's a sample and binding buffer. It's just small words. You open it and you drop there a small piece of sample that you like to take DNA out, yeah? Extract the DNA. That's just a half gram. It's like, think about 5 mm by 5 mm by 5 mm cube. Very small one. Like half gram to 1 gram. You just drop it there and then wait until it's completely dissolved. And after it's dissolved, you take those things. It's special columns for purification of the DNA. So we have a column and then a collection tube. Yeah. So we put that released DNA, those liberated DNA from the spider or from a tree, from the leaf, from the cow, from the pig, from the goat, from whatever you like, rabbit, rat, here, and then we need to spin it. For the spinning, you can make a self-made, the biohacker style a rotor, where you can put it there, and then you need to connect it somewhere that with a motor that can move it around. Yeah, that's called a centrifuge. So the water, yeah, so the liquid will go through, and the DNA will stay here on that white matter there. It's called filter. And then you add more and do it again and again, and then you take a wash buffer, and then you wash the filter with the wash buffer to remove all impurities, and then you take another one. It's called collection tube. You put this thing into collection tube, and you add their elution buffer. In the elution buffer, the DNA will goes off that white substance and drop there, and so you at the end you will get something like this. This is my number 9. It's my Japanese spider DNA already prepared for next step. It's also possible to use. It does need to be centrifuge. It's also possible to use those devices. You can just use a sucking action, connect it and move it to get it through. Yeah, it's like a vacuum. Yeah, and the last moment when you need to elute it, you just push it from other side into the bathroom. It's just another way to do things. Yeah. And well, I also provide some pipettes for the process. Yeah. Yeah. So spider. I also provide some cuts if you want to cut it. Yeah. And the syringes. Yeah.
Yeah, take 2 hours.
Yeah, no problem. And the syringes. So we have the full set of things to make your own DNA or to use one of those that I already pre-made for you. So now something is really special going to happen. It's called modification, human modification. Yeah. It's a genetic human modification with the spider DNA. So I'm going to be the first in the world legally Spider-Man. What I'm going to do, I'm going to take a small syringe, sterile of course, single use, and take that DNA inside. We don't need to take it all, just small amount. So that's quite a lot of DNA. It's enough for whole family, probably people at least, or maybe for 3 wives. First, I need to make a circle where I want to do it. Yeah. I want to do it, let's say here. Yeah. It's nice to have a tattoo in that area with the shape of the animal you like. And yes, put it here. And just under the skin, slowly, very slowly, go in and inject it. Simply, easily, without any pain, is going in. So now I have my spider DNA in my body. It's here. Next step is electric stimulation. It's a 1000 volt, very short, 3 times short of their it's here. Yeah, it's now only charged. So can I get it here? So I need to deliver the electric pulse to the place of injection. 1000 volt. Yeah. So it's going to be 3 pulses there. So it's not it's a charger. the electric is completely different there. It's electroparator. Yeah. It has a step-up transformer and rectifier bridge and capacitor. Yeah, and also some small electric chip that provides some intelligence to that process here. Okay, so now we are going to do 1000 volt electric pulse, electric stimul to the area of injection. It's 1 electrode and that's it. It's all the electric board is doing. And voila, that's what we have. Now I'm officially Spider-Man. What is going to happen with that DNA? Of course, most of the DNA is going to be destroyed by the body. But because of that electric impulse, some of cells will become very curious, those fibroblasts and fibrocytes, and they will take it in. In many cases it will be also disappear, but in some cases it will be taken inside of genome, inside the nucleus of the cell. And there it will be incorporated into the genome. It's called non-homologous recombination. And those cells will take care of the DNA, and they will reproduce it, and daughters of those cells, they will also carry those spider DNA. So legally I'm a Spider-Man, I'm a chimeric organism. Now, first in the world, the real Spider-Man.
Okay, sir.
In Brussels, yeah.
Okay, sir. At the end of the interview, so now you're Spider-Man, but where is Spider-Woman?
Yes, we need just to inject another someone.
You need to inject a
Spider-Woman. If I want to make a spider child, for example, I need to inject it not here, but in another place of the body. And it's also possible. It's called SMGT. It's a sperm-mediated gene transfer. It's also possible, yeah, but it exists. It's scientific stuff. It was successful. There is a lot of publications about it. So, but I need to inject it in another place of my body, and to deliver also electric impulse there, which is pretty much a special perspective for the most of people to think about.
To kind of end the interview, I'm not kind of interview since 20 minutes or something like that. But you are telling very good explanation about crazy thoughts and doings. But my question is, what are you doing with the clothes on in the bath?
Just because of good light. It's a very nice light here. I mean, the light. It's a perfect place for such filming. Because where else people can believe in this? It's too much real to believe in that. So I need to be strange. So naturally, it's a difference between the dream and reality. What's the difference between dream and reality? Well, where I'm now, I'm in a dream or in reality. And the people who will watch it, they are going to see a difference between dream and reality. They will believe that I'm in a dream. I'm from the Hollywood movie. But it's not. It's a perfect. Look at this. I explain you how it works. It's a science. It's just usual science. There is nothing anti-scientific in it. Yeah. Everyone can do it. You don't need to have a lot of experience, or you don't need to study 5 years biology and get PhD and 15 years of experience. You can just do it with my kit. There's very simple instructions. Everyone can become a hybrid. No, it's not hybrid. It's chimera. Yeah, the better word to use it is chimera, but I use hybrid here. Join the army of hybrids. You can make connection, that special connection with everything. I have a potato DNA there. Let's say next time you eat potatoes, if you did it with potato DNA, you can talk with potatoes because they are your friends. Their DNA is living in you. The DNA of potato alive, and it's propagate inside of you. The major question you can ask, like, what's the dangers of it? Yeah, like, is it good? Is it safe? That is the really big question. How safe it is? Because there is only 3 scenarios. First one is nothing will happen. Second one, scenario that
cells take it in and incorporate, and again they will just keep it wondering about it, talking with that spider DNA and making some friendship, yeah. And the worst case scenario is that, you know, spiders are very old. They 200 million years old, and there's some ancient virus from that who was sitting quiet all those 100 million years in the spider DNA. When he gets into human environment, he will become really excited, like wow, like a genie from the bottle. Yo, new life, and it will be resurrected. And I will be patient number one who will start a huge epidemic on the planet that will wipe maybe 95% of humanity. From my point of view, it's not that bad, really. Humans are growing very, very fast, and we can make those 5 billions back in no time, just in 100 years. They will be back 5 billions, even from 5% remaining. And maybe it will be better humanity after all.